Hi. So, in our last discussion, we looked at the various ways in which reproductive isolation can happen between groups or species, and then we asked the question that how exactly can these mechanisms of reproductive isolation evolve. Now, why is that a big question? That is a big question because reproductive isolation requires that some individuals will either not mate at all, or even when they mate, they are going to be, the offspring are going to be infertile, or the offspring are going to have lesser amount of fitness. Now, understand that when such a thing happens, then the fitness of both the individuals who are taking part in reproduction as well as the entire population drops. Now, all else being equal, organisms, when they are freely exchanging genes, and selection when it is trying to maximize the fitness of the individual, then such a situation, or rather anything that reduces the fitness of the population or the individual, is not going to be promoted. So, if that be the case, how exactly will reproductive isolation mechanisms evolve? So, that is not a very easy question, and frankly speaking, the answer is that somehow the ability to exchange genes have to be reduced. Once it starts getting reduced, then all kind of other mechanisms can happen, but at the first step there has to be some reduction in the ability to exchange genes. And the simplest way by which it happens is some kind of a geographical isolation. And geographic isolation can happen in multiple ways. The most well studied one is what is known as allopatric mode of speciation. Now, what is that? Now, allopatric, the word allo means other, and patric refers to pater, which basically refers to homeland. So, it is like other homeland. So, this is the case where the formation of a new species happens because a single population somehow gets subdivided into two or more geographically isolated populations. Now, how exactly can that happen? Now, it can happen in two major ways, either if you have what is known as vicariance or if you have dispersal. So, what are these things? So, vicariance is the scenario when you have a population. So, let us look at this example. Suppose you have an original population and somehow a geographical barrier rises in the somewhere inside the population, within the range of that population, and the geographical barrier can be in the form of a mountain range or a river or some kind of a upliftment of land, or somehow the land going down and forming some kind of a valley or whatever, or in many cases sea level changes. So, when you have a geographical barrier coming up like this, then populations which are on either side of the geographical barrier, they are no longer able to exchange genes. And when that happens, then the two habitats, they might end up becoming slightly different. Selection might lead to different outcomes. There can be different kinds of mutations that might come up in these populations. Ultimately, all of this will lead to reproductive isolation, such that at some point from one species you will end up having two different species. So, there are quite a few well known examples of vicariance. I am just giving you one. So, you know that you have North America and then you have South America, and these two continents are connected by a land bridge which is known as the Isthmus of Panama. That is what I am showing you over here. Now, turns out that the Isthmus of Panama did not exist at all times. So, there was a time when the Atlantic Ocean and the Pacific Ocean were actually connected at that place, and then about 3 million years or so ago, the Isthmus of Panama slowly rose from the from below the sea. And when that happened, when it formed a landmass, then the Atlantic and the Pacific Ocean got separated from each other, and as a result of that, the environment on the two sides actually changed. So, there were differences in terms of tidal fluctuations, water temperature, the clarity of the water, how much of salinity was there on the two sides. All these factors varied across the two sides. So, obviously, the species which earlier were continuously distributed on both sides of the isthmus, now they became separated and they started evolving. differently. And this actually has been observed in multiple taxa, but I am going to show you in the context of a taxa where it has been studied very well. These are known as the snapping shrimps. The genus is Alpheus. Now, this is a very interesting organism. So, you can see this one limb of its highly enlarged. So, what it does is it has a mechanism over there using which it is able to compress air and then release it at a very high pressure, and as a result it is almost like a bullet of water that goes and strikes prey and stuns it. Okay, it is not a bullet per se, it is more like a pressure wave that goes and strikes the prey and stuns it, and in the process there is a massive snapping sound that is created, because of which these are known as the snapping shrimps. Now, turns out that there are many species of this particular thing, but there are 7 pairs of sister species. So, what are sister species? Sister species are species which are very, very closely related to each other. Okay, the most closely related species to each other in some sense, and there are 7 such pairs on either side of Atlantic and Pacific. And turns out that if you look at these sister species, at the genetic level there is quite a bit of similarity, but because of this vicariance event, they have ended up now becoming reproductively isolated from each other, and even if you bring them together and in nature or under laboratory conditions, they are not going to be able to mate. So, this is one way of achieving allopatric speciation. The other way of achieving allopatric speciation is through dispersal. So, what is dispersal? So, basically when a small group of individuals migrate to a new isolated habitat and thereby effectively creating a new geographically separated population, that is when you have the dispersal mode of allopatric speciation. So, for example, when you have, let us say, an island being colonized by some organisms from some place. So, this is exactly similar to the vicariance mode that we saw, except that instead of having a specific geographical barrier in between separating the two subpopulations, the barrier essentially is distance, just the fact that they are quite a bit away from each other, that is it. Now, again there are a few very well known examples of this, and the one that I am going to share with you is related to an island somewhere, or not an island, it is actually a group of islands about 1100 kilometer west of South America. I am sure you can recognize which is that is about the famous Galapagos Islands. So, this is where Darwin went and saw his tortoises and more importantly the finches. So, if you look at the Galapagos finches, there are about 18 species of those finches. They are also known as Darwin's finches, and I am showing you just 4 of those 18 species over here. And what is more important is the fact that all of them are supposed to have descended from just one migration event which happened about 1 or 2 million years ago, and that migration event happened from the mainland, and after that there were multiple speciation events. But the main thing that I am talking about is the fact that the original thing was a dispersal event, which then subsequently led to all these 18 species or so. Now, instead of giving you the details of this myself, I am going to request you to watch a particular video. This video again is one of the HHMI videos. This is about the work of Peter and Rosemary Grant, who spent more than 50 years studying these birds in their natural habitat on the Galapagos Islands. So, this is a very, very famous piece of work. As a matter of fact, this is one of the most well known pieces of modern work in evolutionary biology, and I strongly recommend that you look at it, not only to understand how new species arise, how the Galapagos finches arose, but also how one can visualize the effect of selection in nature and see selection work right in front of one's eyes. And after that, I also want you to look at one more video, which is this one, wherein On the Galapagos Islands, the grants were able to document the birth of a new species, interestingly again due to a migration event. And so these were the so called big birds, and I do not think they have named the species yet, but what happened was that one bird migrated into the Galapagos Islands and it had a very different song, therefore, it should not have been recognized by the existing species over there. But somehow there was a freak female on the Galapagos island, female finch, which ended up mating with this migrant finch, and somehow the babies that were produced, they were all fertile, and then they have ended up establishing a new species, a new population. So, have a look at this particular link that I am showing you at the bottom. Also look at the there is a video over there, see that video, but there is also a write up over there which describes in very simple language what exactly these people saw. Absolutely brilliant stuff. So, that was about the dispersal mode of allopatric speciation. Now, the question is, how exactly does allopatry lead to reproductive isolation? And this is roughly speaking what happens. Once the subpopulations get isolated, then gene flow ceases, there is no more migration, and after that these separated populations, they start diverging. Why do they diverge? They diverge because of a bunch of things that can happen together or separately. So, first of all, the environments are slightly different, as I showed you in the case of the Isthmus of Panama arising, and because of the environments being different, the selective pressures in the new environments are slightly different. Of course, they are now evolving independently. So, the mutations that arise and rise or go down in frequency, they are also different, and more importantly, particularly if the populations are small or smallish, there is a effect of genetic drift, and as you remember, genetic drift, the effect of it cannot be predicted, it is quote unquote random with respect to fitness. So, because of that also the populations end up diverging, and over sufficient time these differences may end up accumulating within the populations such that it might lead to reproductive isolation. Now, what kind of differences are we talking about here? And basically, all the mechanisms that we dealt with in the context of both pre-mating and post-mating, and pre-zygotic and post-zygotic reproductive isolation. So, for example, the structure of genitals, they can end up diverging. There might be behavioral differences, for example, in terms of courtship song or courtship dance or whatever, mating rituals and so on. There can be biochemical differences, or as we pointed out, due to selection, mutation, etc., the differences might even be at the genetic level. So, the other three things we have already dealt with in the previous lecture, but I will just say a few things about what kind of, or rather how differences at the genetic level can potentially lead to reproductive isolation. So, what I am going to talk about is known as the Dobzhansky-Muller model. Dobzhansky is Theodosius Dobzhansky, Muller is Hermann J. Muller. We have met both of these people in our previous discussions. So, what they suggested was that suppose you have an ancestral population. This is a highly simplified cartoon kind of a model. So, suppose you have an ancestor, and let us say the ancestor has two loci, and let us assume that the ancestor's genetic makeup is A1 A1 B1 B1. So, capital A1 A1, capital B1 B1. Now, at some point there is a geographic separation that happens, dispersal vicariance whatever, and again population 1 is A1 A1 B1 B1, and population 2 is also A1 A1 B1 B1. But now, let us assume that genetic divergence has started happening, because of which let us say that in population 1 the A1 allele gets replaced by the A2 allele, whereas in population 2 the B1 allele gets replaced by the B2 allele. Okay. Now, because of this, what is happening? The A2 allele, it only evolves in presence of B1, and the B2 allele, it only evolves in presence of A1. Okay. So, B2 has never met A1 and A2 has never met B1, right. Now, because of this, the epistatic effects might be such so that when suppose for whatever reason now these populations are coming back in contact with each other and they are hybridizing, then you some of the hybrids they are going to have A1 B1 or A2 B2 coming in conjunction with each other. And because A2 has never evolved with B2 and B2 has never evolved with A1, there can be potential negative epistatic effects when these alleles come into contact with each other, which might end up reducing the fitness of the hybrids. So, this is what it works, how it is described, that DMIs, Dobzhansky-Muller incompatibilities, describes the negative epistatic interactions that occur between different alleles of different genes with a divergent or different evolutionary history. Now, do we have any reasons to believe that such a thing even works? Do we have any examples? Of course, we do. So, I will give you again just one example. So, this is about this species of Mexican swordtail fish, where the genus is Xiphophorus. Okay. So, these two species they diverged from each other about 250,000 generations ago, and turns out that these are the two specific species we are talking about, Birchmani and Malinche. Now, if you look at the Birchmani species, some of them, not all, some of them have a spot, a black spot here on the tail. Okay. Malinche has nothing like that. So, you can see they are physically or morphologically they are very different from each other. Now, when you mate these two species, you get a hybrid. Then some of the hybrids they are going to inherit the allele for this caudal spot. Now, turns out that this caudal spot it actually has zero effect on the fitness of the Birchmani. However, when the caudal spot allele finds itself in a hybrid, then in a rather high proportion of the hybrids, it ends up developing malignant tumors, and people have actually figured out which is the gene which is responsible for this. This is due to a gene which is known as the XMRK gene. Essentially, it is a melanoma receptor tyrosine protein kinase. So, here is a situation where this particular allele of this gene is perfectly benign, harmless when it finds itself in the Birchmani background, but the moment it gets into a hybrid background with some genes from Malinche, then it ends up causing the tumor and thereby greatly reducing the fitness of the hybrids. So, we talked about allopatric speciation, I give you some examples, but those might be specific cases. Do we really have evidence to believe on a large scale that allopatric speciation can generally occur, and essentially what I am asking is, can distance or separation be a major factor in terms of organisms diverging from each other? And turns out that we do have a couple of predictions which have very nice empirical support for them. So, we will look, well, actually there are quite a few, I am just giving you two of them. So, let us first get hold of the logic, then we can understand what exactly is being done over here. So, if geographical isolation plays a big role in creating reproductive isolation, then populations of the same species that are further away from each other, we will predict what? We will predict that such populations should be more differentiated than populations that are close by. Why? Because the further away you are from each other, the lower is the chance of exchanging genes, and the lower is the chance of exchanging genes, the more differentiated you are going to be, and therefore, obviously distance and differentiation will be positively correlated with each other. Do we have any evidence to believe this? So, in order to check this out, Tille et al. They investigated the mating behavior of pairs of wild salamander populations, some name Desmonathus ochrophius. separated by different geographical distances. So, what they did was they took males and females from many populations which were separated from each other by different distances. Okay. So, what they did was it is a humongous experiment. So, they took males and females from all these populations, brought them to the lab, and then essentially mated males of one population with females of different populations, which all of which are coming from different distances, and vice versa, which means kept the female constant and then gave them males from different populations. So, essentially they created bunch of mating pairs where the males and the females were from populations which were geographically separated to different degrees, right. And then they tried to see in what fraction of the cases mating is happening. So, so this is the species Desmonathus. What you have here on x axis is the geographical distance in kilometers between the populations, and what you have on the y axis is sexual isolation index. So, basically the more the sexual isolation index, the lower is the probability that they are going to mate. And what you can see is a very clear positive correlation between the two. The more the geographical distance, the less the greater is sexual isolation, which means that the less likely they are likely to meet. So, this shows us that what we predict from allopatric speciation, that distance should be correlated with isolation, it looks like we have a validation for that. So, another prediction that can be made in the context of allopatric speciation is in terms of the island populations. Now, we are saying that allopatric speciation depends on gene flow. Okay. Now, of course, gene flow in many cases is correlated with the dispersal with the distance, but it is also related to how dispersive the species is. Okay. So, suppose let us say you have a snail on one side and let us say you have a bird on the other side. Obviously, the distance that the snail can move and therefore, the distance that the is required for gene flow is very different, much smaller for the snail compared to let us say the bird. So, now instead of talking in terms of distance, we are going to talk in terms of gene flow, which explicitly takes into account the dispersal propensity of the organisms. So, the idea is that if you talk about a continent which is a huge landmass, then of course, it is difficult to figure out who is going from where to where and how speciation is happening. But if you think about island populations, then you can be more sure that if you have two sister species, then they have essentially come arisen from a common ancestor, because it is an island population, right. So, it is very less likely that they would have come from a different place. So, I will qualify that as I go forward. So, the point over here is that if you have island populations, then species which have greater gene flow, they should require more area for speciation, versus species which have less amount of gene flow, they should require relatively less area. So, what Kissel and Barraclough did was they looked at different pairs of sister species, and through genetic mechanisms etc., they made sure that they are species which most likely have originated on the islands, and then they essentially asked the question that if they also, using genetic methods, they figured out the rate of gene flow. We did not discuss it over here. There is something known as FST, through which you can get the measure of gene flow. So, they asked the question that what is the minimum area required for a sister species to be found. So, what is the smallest island on which, let us say, I have found a snail to speciate? What is the smallest area of the island in which, let us say, I have found a bird to speciate? So, that is the data that they got by crawling through an enormous amount of literature, and that is what has been plotted over here. So, on x axis we have 1 by FST, which is essentially increasing gene flow going from left to right, and on the y axis we have the minimal island size in which a speciation event has been observed for that group of organisms. And they have taken snails, and these are flowering plants, small birds, these are lizards, ferns, insects, mammals, and these are specifically bats. Now, what you can clearly see is that there is a very nice positive relationship. In other words, the minimal island size which allows speciation is small in taxa which have limited gene flow like snails, etc. Whereas, if you look at those species which are able to migrate a lot more and therefore, in general have a greater gene flow, they require much larger islands in order to speciate, showing that gene flow is going to be related to the rate of speciation. So, that was about allopatric speciation. So, there is a special case of allopatric speciation known as peripatric speciation. Okay. So, what is the special case? So, normally when you talk about allopatric speciation, then people think that the populations which are getting separated, they are reasonably large, okay, not too small, let us say. But there can be a special type of allopatric speciation where the new species are arising from small isolated populations which are on the periphery of the larger ancestral species range. In other words, we are talking about very small populations which geographically are not very far away from the ancestral range, but there is some kind of a barrier nonetheless. So, the main thing over here is the fact that one of the populations, the subpopulation, is going to be much smaller than the main one, such that genetic drift actually is what is going to play a major role in this speciation. So, in the other case, in the allopatric case, typically it is mutation, selection, those are the ones that are supposed to be more important. Drift plays a relatively less role. In this particular case, drift is supposed to be a major mechanism. So, as you can see, this is not a very clean distinction or anything, which is why peripatric speciation, many people do not even talk about it separately. They simply say that okay, this is just allopatric speciation one more type, that is about it, and that is how you should also look at it. So, this geographical isolation that we are talking about, it can happen in 3 ways. What are the 3 ways? One is that through dispersal. So, you have the original population, a small number of those go and settle somewhere close by to the original range, but after they go there, dispersal more or less stops, because of which this small isolated population on the periphery or the range of the original population, this one diverges and it ends up forming a new species. The other way in which it can happen, which is what is known as centrifugal speciation, is where you have the original population and then for whatever reason the original population expands its range. Okay, so, the members are now found over a larger area, and then for whatever reason, bad environment or whatever, the range again contracts back, but while it contracts back, it leaves a small subpopulation somewhere within the broad range area of the expanded range, but outside the original range. And again there is no gene exchange between these two subpopulations, and at some point speciation ends up happening. So, this is centrifugal speciation. And then the third one is exactly like the vicariance situation, wherein you have original population and within the range of that some barrier is created, because of which one little population gets separated out, and then this little population speciates and becomes a new thing. So, as you can see, it is conceptually extremely similar to allopatric. Now, because of this, it is very difficult to distinguish between an example of peripatric speciation and allopatric speciation, So, one way you can do is you can think about what is the signature of drift in the genome. But then the problem is, how do you know that it did not speciate allopatrically in large population size and then subsequently underwent drift, versus it speciated right at the beginning with a small population via drift. So, these are frankly speaking tough things to do, and even the example that I am going to show you, it is reasonable, but it is not absolutely 100% foolproof. So, this is an example which is related to this tree, species of tree known as the Picea species. These are spruce trees, and these are found in many places, but I am primarily going to talk about spruce trees in North America. So, there you have two different species of Picea. One is known as Picea rubens, the red spruce tree, and the other is known as Picea mariana, the black spruce tree. So, as you can see that these are conifers. So, the shape and the color of the cones are very, very different from each other. Now, people have done a lot of phylogeny and genetic studies on that, these two species, and they have figured out that the geographical range of the red spruce tree is actually much smaller than that of the black spruce tree. Okay. And if you look at their sequences, you realize that there is a very high degree of genetic similarity between the two taxa, which is suggesting that they are very closely related to each other. But if you look at the differences, genetic differences between the two, you realize that the genetic diversity of the red spruce tree is actually far, far lower than the black spruce tree, both in terms of nuclear as well as mitochondrial DNA. So, this suggests that most probably this red spruce tree kind of came out of the black spruce population like a founder event. Also, the red spruce possesses no unique mitochondrial haplotypes. Everything that it has is a subset of the black spruce mitochondrial haplotypes, which again suggests that it is like a small subset that broke off from the black spruce tree. Thus, all these evidences together are suggesting that the red spruce speciated peripatrically from a black spruce population, and based on how much divergence is there, people have estimated that this most probably occurred due to glaciation, because of which a small subpopulation of the red spruce ancestor got separated out from the main black spruce population. Great. So, we talked about the first mode of geographical isolation, allopatric speciation, and a special case of allopatric speciation, which was peripatric speciation. So, now we are going to look at a second type of geographical isolation, something which leads to a mode of speciation known as parapatric speciation. So, what is it? So, in this particular case, there is reproductive isolation happening between the two populations, but there is geography involved, but there is no barrier, and these populations are actually exchanging genes, but they are not doing so freely. So, what do I mean by all these things? So, essentially parapatric speciation occurs when the populations are continuously distributed, but the divergence happens because there is reduced gene flow, and much of the time this reduced gene flow happens because there is an environmental gradient over the geographical range. So, because of this gradient, in different regions of the geographical range, selection operates on separate things, and because of that there is lack of gene flow. So, just to give you a diagrammatic representation of the whole thing, let us assume that we have time going from left to right. So, you have the original population, and then for whatever reason a cline gets set up, an environmental gradient gets set up, and when the environmental gradient gets set up in different regions, different kind of things, different suit of traits are getting selected for, because of which you have let us say original traits in one area, new traits in another area, and this is the important bit, there is going to be an area somewhere in the middle where the hybrids are going to be around. And over time the fitness of the hybrids, if they are reduced compared to both the sides, then over time these hybrids they end up having lower fitness and they just disappear, because of which speciation ends up happening. So, the main thing that is happening over here is that this is predicting that there will be hybrid zones in a large fraction of times between the two populations, where the two speciating populations meet. So, do we have evidences for this? Okay. So, there will not be any barrier, there will not be any distance, but because there is a change in the environmental gradient, because of which there will be low fitness in the middle, and because of that speciation will happen. That is the whole idea. So, just to say that in more concrete words, unlike allopatric speciation, no absolute physical barriers completely preventing gene flow. Populations are still adjacent to each other, they may still interbreed at the borders, but it is this gradient that is creating distinctive selective pressures in adjacent areas, and individuals will typically mate with their geographical neighbors, and because of that there is going to be limited gene flow where the gradient is changing, the hybrid zone so to speak, and when these diverging populations meet, the hybrid zones will be formed, but these hybrids will end up having reduced fitnesses because their traits are neither well suited for this side of the environmental gradient, nor is it well suited for the other side of the environmental gradient. And it is this reduced fitness in the hybrid zone which will reinforce the selection against interbreeding, and therefore reproductive isolation is going to evolve. Slightly complicated, but let me show you the example, hopefully that will make it clear. So, this example is from the mines in UK. So, you know that whenever mining happens, then the soil in the neighboring area gets thoroughly contaminated, and in this particular case the soils were contaminated with very high levels of heavy metals, specifically zinc and lead, and this had happened for about the last 100 years. So, the soils were in really bad condition, but near to them there were pastures, and those pastures the soils were reasonably good. So, turns out that there was this species of grass known as sweet vernal grass, and these grasses, some populations they adapted to grow on the highly contaminated areas, whereas other populations of the same grasses they were growing on the less contaminated areas. So, these populations that adapted to grow into the contaminated areas, they actually changed quite a lot, and more specifically what changed was their flowering time. So, these metal adapted populations they were flowering much earlier than the non-metal tolerant plants, and as a result of this there was a very strong pre-zygotic isolation that happened between the metal tolerant and the non-tolerant populations, and this pre-zygotic isolation has been maintained for more than 40 years. So, just think back, all the cases that we discussed till now, there was geographical isolation that happened, and that led to the evolution of reproductive isolation. But then what happens if the geographical isolation breaks down? Okay. Maybe the river which had started flowing, it again changes its course, and now the populations again come in contact with each other, or maybe the populations have again done some range expansion due to whatever reason, and in another area of their range now again they have come into contact with each other. What is going to happen when there is, this is what is known as secondary contact. Okay. So, what is going to happen if there is secondary contact? So, what are the possible outcomes of secondary contact? One outcome is maybe reproductive isolation has not really evolved. So, when there is secondary contact, they again freely exchange genes with each other, and then again they meld back to form a single population. So, as if the entire divergence has not really happened, they are back to the square one. Or what might happen is, if some amount of divergence has happened which has made one population more adapted or more competitive than the other, then the two populations might end up competing with each other, and one population might drive the other population to extinction. This is what is known as competitive exclusion. A third possibility is that, like what we were talking about in the context of parapatric speciation, when these two populations meet, the diverged populations meet, then they hybridize in the zone where they are meeting, but there is reinforcement of the reproductive isolation simply because the hybrids have lower fitness. So, in other words, everybody from one population who has a tendency to mate with the individuals of the other population, their fitness goes down, and therefore, the tendency to avoid the other population, that is what gets selected for, which ultimately obviously leads to reinforcement of the reproductive isolation. Or the fourth thing that can happen is that the populations meet, the fitness of the hybrids is low, but that does not lead to reinforcement. In other words, the two populations, they just maintain their distinctive identity except for a narrow zone in the middle where the hybrids are there, with some mild gene flow happening over there, but rest of the identity remains as it is. So, till now everything we talked about was speciation in the context of geographical isolation. But can we have speciation without any geographical isolation? And turns out that we indeed can. And this is what in technical language is known as sympatric speciation. So, allopatric I said was other homeland, sympatric is same, patric homeland, so same homeland when they are in the same geographical area. So, how can this happen? So, suppose you have an original population and then something changes inside the population in a subset of the population, due to which, although they are in the same geographical area, one group stops or reduces exchanging genes with the other group. And when that happens, that is when you have the formation of a new species, even though the two groups are still in contact with each other, and this is what is sympatric speciation. Now, obviously, what we are trying to say is that there is reduction in gene flow, but that is happening in the absence of geographical barriers. So, what kind of factors can lead to that? Turns out there are three major factors. One we have already discussed is polyploidy. Polyploidy is when the entire chromosomal number changes, and although it is somewhat uncommon in animals, it is actually very common in plants, where it accounts for about 15% of all the speciation events. The other thing that can happen is sexual selection, where the males or the females preference for certain traits can, in one group can make them very different from another group which does not have that preference. And the third thing, of course, is some kind of a habitat differentiation or resource partitioning, where the environment in which the organisms exist, some individuals specialize to in one part of the habitat, some individuals specialize in another part of the habitat. We will look at examples of all three one by one. So, example of polyploidy in plants we have already dealt with, remember the speciation in the primroses in Kew gardens, Primula cubensis, but that was something that had been artificially induced. But as it turns out, polyploidy in plants is very common under natural conditions. So, we will look at one very famous example. So, there is a species of plant known as the goat's beard. So, in early 1900s, 3 species of this goat's beard were introduced into North America from Europe, and for this all these 3 species, the haploid species number n is equal to 6. So, what species are these? So, these are the 3 species called Tragopogon dubius, Tragopogon pratensis and Tragopogon porifolius. Now, around 1950s, people, as in the scientists, they suddenly started seeing 2 new species of this particular plant, Tragopogon. And they were like, where the hell did this come from? And when they looked up at the chromosomes and at the molecular level, they realized that one of them, Tragopogon miscellus, had actually arisen due to a hybridization between pratensis and dubius, and the other one, T. mirus, had arisen due to a hybridization event between dubius and porifolius. So, essentially these two, they have n is equal to 12, which is double of what was happening for the 3 parental species. So, this is allopolyploidy, where you have 2 different organisms, 2 different species coming together to lead to a polyploid hybrid. And as a result, this is also an example of hybrid speciation, where 2 different species come together and in one step form a new species, which is not fertile, reproductively isolated with the parental species. So, this is sympatric example number 1. The other way in which sympatric speciation can happen is through sexual selection, and we are going to talk about this particular lake called Lake Victoria. This is one of the largest lakes in the whole world. As you can see, it is somewhere in eastern or central eastern Africa, and it has many claims to fame, but one of its major claim to fame is that it has an amazing fish diversity. So, if you look at this particular lake Victoria, there is a particular kind of fish called cichlids. It has more than 500 species of cichlids alone. Okay. Now, we are going to come back to this 500 species of cichlids later on, but right now I am telling you a different story. Now, today unfortunately the number of species has gone down quite a bit in Lake Victoria. That's because humans at some point decided to introduce a new fish called Nile perch over there. That fish turned out to be invasive and extremely aggressive and ended up just outcompeting many of the existing cichlids, but we still have quite a few. Now, among the ones that are still alive, there are 2 existing species which are very, very closely related to each other, but I mean in these species the females, they look very, very similar to each other. You will be very hard pressed to distinguish them based on morphology alone, but if you look at the males, they look very different. How different? So, let me just show you. So, this is Pundamelia and you can see that it is bluish grey kind color, and this is Pundamelia nereri. Okay. So, this, as you can see, is very, very brightly colored. Now, turns out that in both these species, the females of the species, they have a very, very strong preference for the males, for the color of their own males. So, in other words, Pundamelia females will only prefer males which are bluish grey in color, and Pundamelia nereri females are only going to prefer males which are discolored. Because of which, although these 2 species are sympatric, in spite of that they are unable to exchange any genes. The females are simply not going to mate with Pundamelia females are not going to mate with nereri females and vice versa. Very interestingly, people have shown that this choice is heritable, and this selection, because the choice is heritable, the selection for this trait is still extremely strong. In other words, Pundamelia nereri females, they prefer males which are more red compared to males of their own species which are less red, which means that there is very strong selection for the nereri to become redder and redder, because of which the divergence from Pundamelia is going to go up even more. So, this is an example where sexual selection is driving reproductive isolation. And then the third example is that of habitat sympatric speciation through habitat differentiation. And the specific example that I am going to share with you is this particular fly called the railroad worm. Of course, the worm refers to its larval stage. It is called Rhagoletis pomonella. So, turns out that this fly in North America, originally it only used to lay its eggs on a particular fruit known as the hawthorn fruit. Okay. But somewhere around 1850s, apple trees got introduced in North America, and when that happened, in some places some of these flies they shifted over to apples, and when that happened they ended up adapting to the apples, and that actually had very interesting consequences. So, two populations started emerging, one that was specializing on hawthorn, the other that was specializing on apples. And how did it work? Now, turns out that the fruit, the host, the apples they mature earlier compared to the hawthorns, and because the apples matured earlier, the flies which were adapting to the apples they shifted their timing of emergence and they started emerging earlier. So, here what I am showing you over here are dates, okay, and what I am showing you over here are number of males that are captured within 1 hour sampling. Okay. So, here is the fly, and this red one is showing you those flies which are specialized on the apple fruit, and you can see the peak emergence of these guys is here in this zone, whereas these are flies which are specializing on the hawthorn fruit. Note that their peak emergence is much later, and more importantly there is very little overlap, there is just a small little overlap over here. Now, you will think that that much is enough for interbreeding, but no, turns out that something else has also evolved. What is that? Which is that the females they have also developed a preference for laying eggs only on that kind of fruits on which they grew up, and also seeking mates only on that fruit. In other words, the populations which have adapted to apple, they will lay their eggs only on the apples, and they will mate with males which are only there on the apples. Okay. So, even if some of the hawthorn flies, male flies are coming out when the apples are still around, they are not going to go there. Okay. And males are also seeking mates on the preferred host fruit, which is why they are not going to go there, and even if they go, the females are going to lay their eggs only on the apples, they will not lay eggs on the hawthorn and vice versa. So, this behavioral isolation on top of this temporal isolation has essentially ended up further reducing the gene flow, and because of which now these two flies have started becoming genetically differentiated. You can find a lot of difference, although it is not yet reproductively isolated. So, if you can bring them together in the lab, they will end up mating and producing fertile offspring, but looks like they are going in that direction. Great. Now, temporal separation is one way by which under sympatric speciation the organisms can lead to reproductive isolation. There is another way which is known as character displacement. Now, what is that? So, let me give you the definition first. So, when similar species or groups diverging populations of the same species, when they live in the same area sympatric, then the differences in their traits become more pronounced. But when their distributions do not overlap, then these distinctions fade or disappear, which essentially means that their trait distributions start overlapping. How does this happen? So, let me just show you through a cartoon. So, let us assume that we have two species, and what you are seeing in green and blue are the phenotypic distribution of the traits. So, in this particular case the trait is the size of the bill. So, let us assume that these two populations they are in totally different areas, and the fitness function it looks somewhat like this, right. Now, when these are in different areas, then the fitness function, even if it is like this in the overlapping zone, it does not really matter for this species. However, when you put them in the same region, then individuals whose bill size are going to be intermediate, they are going to face a problem, and because of which they are going to diverge away, they are going to differentiate away their trait distributions, and the trait distributions are not going to be overlapping to that extent. So, I know this is probably a little confusing. So, let me explain the same thing to you with an example. And prior to that, this character displacement can be another way to reinforce reproductive isolation after allopatric speciation. So, how does that work? Let me explain to you with an example. And the example is in the context of the Galapagos finches again. So, here we have two species. This is called Fulginosa, this is called Fortis, okay, the ground finches, and these are two islands, Los Hermanos and Daphne Major, and what we are showing you are the distribution of the bill sizes on Los Hermanos, where only Fulginosa works, is found, and for Daphne Major, where only Fortis is found. And bill size is very important for these birds, because the bill size determines what kind of food they can eat and with what efficiency. Now, observe the fact that when the birds are found on their own, then there is a substantial overlap between the distribution of their bill sizes, which means that they are essentially eating whatever resources they want to eat, and accordingly their bill sizes have evolved. However, now you look at the same two birds, but now you see them on an island called Santa Cruz, where both birds are found together. Now, when that happens, note that the overlap between the distribution of the two bill sizes has actually almost disappeared. There is a very little overlap. Essentially what is happening is, of course, this is assuming that, and it is known that the resources are similar across all the three islands. So, essentially what is happening is that here the when they are found singly on an island, they are eating whatever resources they find it best to eat, most physiologically suitable for them. But when they are being forced to be together, then they kind of partition away the resources, and one set of resources Fulginosa is feeding on or using, another set of resources Fortis is using, because of which the fitness function is such that they have ended up completely separating out their trait distributions. So, this is what is known as character displacement, and the one sentence summary of this is that the distribution of bill depths, they overlap in allopatry, which means that when they exist away from each other, but not in sympatry, which means when they are together. So, this is another way over which is different from the temporal isolation way, in which sympatric populations might end up separating out their niche, which then might end up having different selection pressure on the two populations, which will reinforce whatever reproductive isolation might have set in due to allopatry. Okay. So, before we finish this topic, remember I told you about Lake Victoria, right, and I told you that I will get back to it. So, turns out, and this is something we have already discussed in the last discussion, there is a phenomenon known as adaptive radiation. And what is adaptive radiation? Remember, we were talking about the fact that different species have different rates of formation of new species, and there is this special phenomena where there is an extremely rapid diversification of a single ancestral species into many new forms, and when that happens in a very small amount of time in a rather small space, you suddenly get an explosion of species. And there are three major features of this phenomena called adaptive radiation. First, as I said, speciation happens very, very rapidly. Second, there is an enormous phenotypic divergence. So, there are many, many different forms or traits that are created. And third, these forms or traits, they are very often specialized to their ecology. In other words, they often end up using different niches in the environment. And one of the so obviously the question that one asks is that what kind of scenarios can lead to this kind of adaptive radiation, and the answers are very simple. Typically this happens when a new resource becomes available or a new habitat gets colonized where there is little initial competition from the existing species. In other words, where the species are free to explore the various possibilities in the environment. And the kind of opportunities that we are talking about, for example, colonization of new island chains. So, if you remember the HHMI video, when we Peter Grant explicitly talks about the fact that there was a migration event into the Galapagos Islands, and because the Galapagos Islands at that point were empty, these birds had all the niches free to them for themselves, and they ended up radiating adaptively into those niches. So, Galapagos Island is a major example of this. Sometimes what happens is that due to geological events or other kinds of celestial events, there are mass extinctions, and when there are mass extinctions, the major competitors are all dead. That is when the guys who got left out, they often end up conquering all those niches or adapting into those niches. And perhaps the biggest example of that is the adaptive radiation of mammals, which happened after the extinction of dinosaurs due to meteorite impact. And finally, sometimes what happens is there are evolutionary novelties. Some groups are able to create a new trait which then allows them to explore and exploit certain niches which were not available to them earlier. So, for example, with the evolution of flight in birds, in insects, in bats, these animals were able to exploit certain parts of the environment which earlier nobody were able to exploit, and once they started exploiting that, then they kind of speciated very easily and very rapidly into those things. So, remember Lake Victoria, we were talking about the cichlid fishes. So, there is this excellent study by Mayer et al. I have taken that photograph from there. So, Mayer et al. Showed that in a span of about 1500 years, which is less than a blink of an eye in the context of evolutionary history, more than 500 species have evolved in Lake Victoria and the surrounding areas. And this, so people have always asked this question, why, what exactly happened? This is the greatest example of adaptive radiation in vertebrates in terms of number of species that is known on this planet. So, these people figured out that actually there were two different lineages of cichlid fishes. One was evolving in the Upper Nile area and the other was evolving in the Congo area, in two different river systems, and somehow they had been doing that for more than 1,000,000 years. And then we are still not sure what exactly happened, but somehow these two lineages, they came together in the Lake Victoria region. I mean, Lake Victoria has a few other lakes around it. So, we are not saying Victoria per se, but that region. So, they came together and they hybridized. And when they hybridized, then this hybridization event, because it led to a massive amount of genetic variation that had separately accumulated in two different areas, that came together, and therefore, this ended up facilitating the subsequent adaptive radiation in that area through recombination and all kinds of other evolutionary processes that we talked about, and that is what led to all these species. I mean, just look at this. There are more than 500 species. I am showing you what, about 10 or 12 over here, but just look at the variation in form, just look at the variation in size, in terms of color, function. There is a huge functional difference between these species. And this is just one group, this is just the cichlid fishes, there are other groups also. So, this is one of the most mind blowing examples of speciation that is known to human beings. The other example that has been extremely well studied and almost as beautiful is in the context of the vangas of Madagascar. So, these are a group of birds. You find them actually, the group is found at different places, but when you look at the vangas of Madagascar, they are a bit special because there are just 40 species. Well, I should not say just to 40, but still 40 species, but there are 21 genera, which tells you that most of these are actually endemic over there, they are found nowhere else, which is true. And if you look at their molecular data and you do the phylogeny, you realize that all these populations arose from one single founding population, just one, okay, and that ended up diverging, radiating into this enormous diversity that you are seeing in front of yourself. And again, this is not complete diversity, this is just a part of the diversity. And very interestingly, all these species, most of them actually eat insects. In spite of that, if you look at the shape of their bills, they are very, very different. Okay, why is that so? Because although they eat insects, the way they eat it is slightly different. So, for example, look at the bill of this one, right? This is a very long and protruding kind of a bill. So, this one actually uses it to probe the holes and other things inside the trees and rocks, etc., and take the insects out. Whereas, look at this guy, look at the fact that the beak is somewhat heavy. So, this one actually strips the bark off and looks at the insects below them. So, although many of them are, almost all of them are insect eaters, in spite of that, the way they eat the insects and from which place they eat the insects, that differs, and because of that their bill shapes, the bill morphologies are very, very different. So, again a superb example of how evolution can lead to endless forms, most beautiful. Now, whatever we have discussed till now, most of the time we have talked about things at the organismal level. Okay. We said that things will happen at the levels of genes, etc., but we didn't really go too much into that depth. But while all these things are happening at the organismal level, at the mechanistic level, at the level of the genes, new things have to happen. New genes somewhere need to come up so that a flightless bird or a flightless organism becomes or develops flight, right. So, how exactly is the genome changing? What exactly is happening at the sequence levels such that we see all these enormous diversification and speciation processes happening? So, that is the next big question that we are going to tackle, but that is something that is going to happen only in the next week. See you then.